Good afternoon, class. Today's lecture is going to be on Unit 4, Chapter 3, Part B. Again, we'll continue with our discussion on cells, and this time we'll focus on active transport mechanisms. Section 3.4, Active Membrane Transport. So the two major active membrane transport processes are active transport and vesicular transport, and both of them require an energy input in the form of ATP to move solutes across the plasma membrane for any of these reasons. So either the solute is too large for channels, or the solute is not lipid soluble, or the solute is not able to move down the concentration gradient. In active transport, a high energy bond, usually in ATP, provides the energy needed to move ions or molecules across the membrane. Despite the energy cost, active transport offers one great advantage. It is not dependent on a concentration gradient. As a result, the cell can import or export specific substrates regardless of their intracellular or extracellular concentrations. Active transport requires carrier proteins or solute pumps, which bind specifically and reversibly with the substance being moved. So some carriers transport more than one substance. So you have antiporters versus symporters. Antiporters transport one substance into the cell while transporting a different substance out of the cell. Symporters transport two different substances in the same direction. Also, active transport moves solutes against their concentration gradient from an area of low concentration of that solute to an area of high concentration of that solute. And this will require an input of energy in the form of ATP. This figure shows the difference between symporters and antiporters. You also have uniporters. Uniporters just help carry a solute across, whereas symporters will carry substance A and B through the membrane with the help of a carrier. Antiporters and an A and B in the same direction, whereas antiporters, A and B would be moved in opposite directions. So notice this figure. You have a phospholipid bilayer with a carrier protein embedded in the membrane, and you have solutes that are moving from an area of low concentration to an area of high concentration. And so to be able to do that, you need ATP or energy, an energy investment to be able to move solutes against their concentration gradients. There are two types of active transport: primary active transport and secondary active transport. In primary active transport, the required energy comes directly from the hydrolysis of ATP, whereas secondary active transport, the required energy is obtained indirectly from ionic gradients created by a primary active transport mechanism. This figure illustrates the difference between primary active transport and secondary active transport. So in primary active transport, the carrier protein or pump will move sodium from an area of low concentration to an area of high concentration, and it's going to move at the same time potassium from an area of low concentration to an area of high concentration. This is going to create an ionic gradient, and notice there's an ATP investment in the primary active transport. Because of the creation of this ionic gradient, now secondary active transport this process will be able to move glucose through the carrier protein. Let's review this concept again with this figure. So in primary active transport, the ATP-driven sodium-potassium pump stores energy by creating a steep concentration gradient for sodium entry into the cell. And in secondary active transport, as sodium diffuses back across the membrane through a membrane co-transporter protein, it drives glucose against its concentration gradient into the cell. And this saves energy because you need an energy input, an ATP input, only for one of those pumps and not for all pumps. we would not stop eating. We would not be able to stop eating if all our pumps, all our carrier pumps would require an energy input. So let's look at primary active transport in a little bit more detail. So the energy from the hydrolysis of ATP will cause a change in the shape of the transport protein. So it's going to cause a conformational change, a three-dimensional shape of that transport protein. The shape will change. Shape change causes these solutes or ions that are bound to the protein to be pumped across the membrane. Example of pumps in the body are calcium pump, the hydrogen or proton pump, and the sodium-potassium pump. So the sodium-potassium pump is the most studied pump. It's the most well-known. Basically, it's an enzyme. It's called the sodium-potassium ATPase. Remember, AS, anything with ASE is an enzyme. And it pumps sodium out of the cell from an area of low concentration to an area of high concentration, and potassium back into the cell from an area of low concentration of potassium outside with respect to inside the cell. So it's located in all plasma membranes, but especially active in excitable cells. So cells like nerves, so neurons, and muscle cells.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
This figure shows the difference in concentration intracellularly versus extracellularly. So inside the cell, intracellular compartments, so picture all cells, have a higher potassium concentration compared to the outside of the cell. And conversely, the inside of the cell has very small concentration of sodium with respect to the outside. The outside, you have a way higher concentration. And so this sodium-potassium pump, what it essentially does is that it will pump potassium from the outside to the inside against its concentration gradient. And it's going to, at the same time, pump sodium from the inside to the outside. Why? Well, to make sure that the inside of the cell always has small amounts of sodium and the outside of the cell has small amounts of potassium.
The sodium-potassium pump is a protein pump found in the cell membrane of all animal cells. Its main function is to transport sodium ions out of the cell and potassium ions into the cell. This serves a number of purposes, but is critically important to the function of neurons as it helps to maintain a higher concentration of sodium ions outside the cell and a higher concentration of potassium ions inside the cell. Preserving these differences in ion concentrations helps to stabilize the cell's membrane potential. This is critical for the neuron to be able to fire an action potential, which is the basis of the electrical signaling of neurons. The sodium-potassium pump is a large membrane-associated enzyme that uses the energy of adenosine triphosphate, or ATP, to transport sodium and potassium ions across the cell membrane. All of the details of how this is achieved are not yet fully clear, but the general process involves the binding of ATP to the pump, which promotes the binding of 3 sodium ions and the release of 2 previously bound potassium ions. ATP is broken down and transfers a chemical group known as a phosphate group to the pump, which prompts the pump to undergo a conformational change, or a change in its shape. This causes the bound sodium ions to be released into the extracellular fluid. At the same time, the pump binds 2 potassium ions, which also prompts another conformational change that returns the pump to its previous configuration, and the cycle begins again. Each cycle of the pump causes 3 sodium ions to move out of the cell and 2 potassium ions to move into the cell. Because there is one more positive charge leaving the cell than entering, there is a net loss of positive ions, which makes the resting membrane potential of the cell slightly more negative.
This is another figure that illustrates the functioning of the sodium-potassium pump. So notice how sodium is being pumped out and potassium is being constantly pumped in, and ATP is hydrolyzed to ADP and phosphate. In addition to the sodium-potassium pump, there are leakage channels that are embedded in the membrane, and as a result, sodium leaks into the cell and potassium leaks out of the cell, and both travel down their concentration gradients. So to prevent what I was talking about, too much sodium inside and too much potassium outside, you have this sodium-potassium pump that works as an antiporter that pumps sodium out of the cell and potassium back into the cell against their concentration gradients. This will maintain the electrochemical gradients, which involve both concentration and electrical charge of ions, and this is essential for the functioning of muscle and nerve tissue. We're going to see this chapter in a lot more detail in Anatomy and Physiology 2. In secondary active transport, so this depends on the ion gradient that was created by the primary active transport system. So the energy stored in gradients is used indirectly to drive transport of other solutes. So the low sodium concentration that is maintained inside the cell by the sodium-potassium pump strengthens sodium's drive to want to enter into the cell. So as it's doing so, sodium can drag other molecules with it as it flows into the cell through carrier proteins, usually symporters in the membrane. So some sugars like glucose, amino acids, and ions are usually transported into the cells via the secondary active transport mechanism. Figure 3.9, secondary active transport is driven by the concentration gradient created by primary active transport. Okay, so on the left side, we see the sodium-potassium pump that is essentially pumping sodium out into the extracellular fluid and potassium into the cell. And this here creates a steep concentration gradient for sodium. And so in the right side of the figure, the secondary active transport, as sodium diffuses back across the membrane through a membrane co-transporter protein, it drives glucose against its concentration gradient into the cell. So glucose is going to move from the outside to the inside following its concentration gradient. In vesicular transport, materials move into or out of the cell by means of vesicles, small membranous sacs that form at or fuse with the cell membrane. And this process also requires cellular energy in the form of ATP. The two major categories of vesicular transport are endocytosis and exocytosis. Endocytosis transport movement is into the cell. There are different types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. In exocytosis, the transport is out of the cell. In this figure, we can see on the top the process of exocytosis, where you have a vesicle from the inside of the cell carrying molecules, and the vesicle will fuse with the plasma membrane, and it's going to empty its contents to the outside. In the bottom figure, you can see the formation of an endocytic vesicle through pinching in of the plasma membrane, bringing in whatever there is from the outside into a vesicle inside the cell. So both of them are what is known as bulk transport. Endocytosis involves the formation of protein-coated vesicles. Usually it involves receptors, therefore it can be very selective. In other words, the substance being pulled in must be able to bind to its unique receptor. And some pathogens are able to hijack the receptor for transport into the cell. They've managed to do that. Once the vesicle is pulled inside the cell, it may fuse with a lysosome or undergo transcytosis. By the way, a lysosome is one of these cellular organelles that is its job is intracellular digestion. This figure shows how you'd have at specific areas of the membrane receptor proteins with that will bind specifically to a target molecule. Then there's pinching in of the plasma membrane and formation of a coated vesicle, and you would have the target molecule coming into the cell. Figure 310 shows the events of endocytosis. So in more specific than the previous figure. So if we follow it with 1, the coated pit ingests the substance. It's typically a clathrin pit, coated pit. So in second step, the protein-coated vesicle will detach, and the uncoated endocytic vesicle, the coat proteins are recycled to the plasma membrane. The uncoated endocytic vesicle is going to join with a lysosome. And the fused vesicle may fuse with the lysosome for digestion, or it can be delivered or deliver its contents to the plasma membrane on the opposite side of the cell. And that is called transcytosis. And the transport vesicle containing the membrane components moves to the plasma membrane for recycling. Cell-eating or phagocytosis is a type of endocytosis. Here we have membrane projections called pseudopods that form and flow around the solid particle that is being engulfed, and the vesicle forms, which is pulled into the cell. The formed vesicle is called a phagosome. Phagocytosis is used also by macrophages and certain other white blood cells, which are components of our immune system. The phagocytic cells move by amoeboid motion. An amoeba is the single-celled organism that moves, using its pseudopods, where the cytoplasm flows into temporary extensions that allow the cell to creep. A small note: macrophages protect our tissues by engulfing bacteria, cell debris, and other abnormal materials. Figure 311A shows phagocytosis. So in phagocytosis, the cell engulfs a large particle by forming a projecting pseudopod. Pseudopod means false foot around it and enclosing it within a membranous sac called a phagosome. and the phagosome combines with the lysosome and its contents are digested. The vesicle has receptors capable of binding to microorganisms or solid particles. So this is something that the macrophage would do to protect us from invaders. The second type of endocytosis, pinocytosis, is referred to as cell drinking or fluid phase endocytosis. Here, the plasma membrane enfolds, bringing extracellular fluid and dissolved solutes inside the cell, and the contents and this vesicle will fuse with an endosome. So it's used by some cells to sample the environment. It's also the main way in which nutrient absorption occurs in the small intestine, and the membrane components are recycled back to the membrane. So figure 311b, we're looking at pinocytosis. So we see here the cell will take a gulp of a drop of extracellular fluid containing solutes into tiny vesicles. No receptors are used, so the process is non-specific. In receptor-mediated endocytosis, this is a selective process. It involves the formation of small vesicles at the surface of the membrane. So it involves endocytosis and transcytosis of specific molecules. Many cells have receptors embedded in clathrin-coated pits, which will be internalized along with the specific molecule bound. So examples would be enzymes, the low-density lipoproteins or LDL that carry cholesterol, for example, iron, insulin, and unfortunately viruses, diphtheria, and cholera toxins may also be taken into a cell this way. Caveolae have smaller pits and different protein coat from clathrin, but still capture specific molecules such as folic acid, the tetanus toxin, and this process also uses transcytosis. Figure 311c, receptor-mediated endocytosis. So extracellular substances will bind to specific receptor proteins, enabling the cell to ingest and concentrate specific substances in protein-coated vesicles. Substances may be released inside the cell or digested in a lysosome. So this figure shows the three examples of endocytosis. So in phagocytosis, a phagosome is formed, at the, using pseudopods. Pseudopods are false feet. So a solid particle is ingested into the cell. Pinocytosis is cell drinking. So, solutes from the extracellular fluid are taken into the cytoplasm of the cell. And in receptor-mediated endocytosis, which is a specific process because it involves receptors that bind to specific molecules or other particles, and these are brought into the cell inside coated vesicles. Basically, exocytosis is the functional reverse of endocytosis. So in exocytosis, a vesicle created inside the cell fuses with the cell membrane and discharges its contents into the extracellular environment. And this is usually activated by cell surface signals or changes in the membrane voltage. So the substance being ejected is enclosed in a secretory vesicle. And so some substances that will be exocytosed would be hormones that are produced by that cell, neurotransmitters, mucus, or cellular wastes. Figure 312a showing exocytosis. So in step 1, the membrane-bound vesicle migrates to the plasma membrane. In step 2, their proteins at the vesicle surface bind with, they're called T-snares or plasma membrane proteins. You really need to not worry about this detail. In step 3, the vesicle and the plasma membrane fuse and a pore opens up. And in step 4, the vesicle contents are released to the cell exterior. Section 3.5, membrane potential. So we're going to do a little bit of neurophysiology here and talk about the resting membrane potential, or also abbreviated RMP. So there's an electrical potential energy produced by separation of oppositely charged particles across the plasma membrane in all cells. And that we know is due to the different concentrations of sodium and potassium and the gradients produced by these two ions. So the difference in electrical charge between 2 points is referred to as voltage, and cells that have a charge are said to be polarized. So the voltage occurs only at the surface of the membrane because the rest of the cell and the extracellular fluid are neutral. And membrane voltages will range from minus 50 to minus 100 millivolts. In different cells, the negative sign indicates that the inside of the cell is more negative relative to the outside of the cell. So in this figure, we see a voltmeter that with an electrode placed just into the cell's interior. And we see a measurement of about minus 70 millivolts. So what this tells us is that the inside of the cell is negative with respect to the outside of the cell. So in this figure, on the left, let's pretend we're looking at a section of a neuron. Remember, neuron are those excitable cells that transmit information to other neurons or glands or muscles. So if you notice, the inside is negative with respect to the inside. In fact, we said that if you measure it with the voltmeter, it would say that it would be around minus 70 millivolts. Now, what contributes to that negative charge of the interior of the cell? So if you remember, the inside of the cell, you have fewer sodium, like a lower sodium concentration and a higher potassium concentration. Also, inside the cell, you have these the A-, these are large proteins. They're negatively charged and they cannot diffuse across the membrane. So how does the sodium and potassium contribute to this? Well, if you look on the right, we have three types of channels. Okay, so we have the potassium channels. I know it doesn't look like a channel, okay, but pretend it's a channel, the green. Okay, so that's the potassium. And so there are more potassium leakage channels. That's what those are, the potassium leakage channels carrying potassium outside of the cell compared to sodium leakage channels bringing in the sodium inside the cell. Okay, so, and this is following their concentration gradient. So if more potassium is leaking out, it means that the potassium is carrying its positive charge outside. Whereas if there's less sodium that comes in, that means that the inside is not going to be as positive compared to the outside. So that's the second reason. Okay, so the first reason is because you have these large negatively charged proteins that are unable to diffuse across the membrane. And two, you have more potassium leakage channels than sodium leakage channels. The third reason is that there's the sodium-potassium pump that for every 2 potassiums that are allowed to get into the cell against a concentration gradient, 3 sodiums are pumped out. So that will also contribute to this imbalance in charge, making the inside more negative and therefore polarized, right? Because there's a charge at this point of the cell. So this figure is also and it's really hard to find a really nice figure that explains this concept. Okay, so what does this figure show? It shows that in the cytosol, there's a greater concentration of potassium and proteins. Lots of proteins are inside the cell. So we said that proteins are not able to, we don't want to lose proteins. So the cell keeps its proteins inside. Whereas outside in the interstitial fluid, we have a greater concentration of sodium. Also, if you look embedded in the membrane, there are more potassium leakage channels. So the membrane is much more permeable to potassium than to sodium. So that means that more potassium is allowed to leak out of the cell following its concentration gradient than sodium allowed to come in. So that contributes to that imbalance in charge. And furthermore, we have the sodium-potassium pump that will pump more sodiums out of the cell, and so 3 sodiums out of the cell for every 2 potassiums into the cell. So potassium is the key player in determining the resting membrane potential. Potassium diffuses out of the cell through the potassium leakage channels down its concentration gradient. And we said that there are more potassium leakage channels than sodium leakage channels. Also, the negatively charged proteins cannot leave. So as a result, the cytoplasmic side of the cell membrane becomes more negative. So potassium is then pulled back by the more negative interior because of its electrical gradient. When the drive for potassium to leave the cell is balanced by its drive to stay, the resting membrane potential is established. And most cells have a resting membrane potential around minus 90 millivolts. So the electrochemical gradient of potassium sets the resting membrane potential. Many cells, sodium also affects the resting membrane potential, because sodium is also attracted to the inside of the cell because of the negative charge. So if sodium enters the cell, it can bring the resting membrane potential up to minus 70 millivolts. So membrane is more permeable to potassium than to sodium, so potassium is the primary influence on the resting membrane potential. Cl- does not influence the resting membrane potential because its concentration and electrical gradients are exactly balanced. Figure 3.13, the key role of potassium in generating the resting membrane potential. So in step 1, potassium diffuses down its steep concentration gradient out of the cell via leakage channels. The loss of potassium results in a negative charge on the inner plasma membrane face. Potassium also moves into the cell because it is attracted to the negative charge established on the inner plasma membrane face. Negative membrane potential of minus 90 millivolt is established when the movement of potassium out of the cell equals the potassium movement into the cell. At this point, the concentration gradient promoting potassium exit exactly opposes the electrical gradient for potassium entry. Also notice in the figure the large protein, negatively charged proteins that are unable to follow potassium through the membrane, further contributing to the negative charge of the cell interior. Active transport maintains electrochemical gradients. So the resting membrane potential is maintained through the action of the sodium-potassium pump, which continuously ejects 3 sodium ions out of the cell and brings 2 potassium ions back inside. A steady state is maintained because the rate of active pumping of sodium out of the cell equals the rate of sodium diffusion into the cell. Neuron and muscle cells upset the steady state resting membrane potential by intentionally opening gated sodium and potassium channels. So this is another type of channel here that we're talking about. These are gated channels as opposed to the leakage channels or the sodium-potassium pump. Okay, so these are different types of channels, 3 different types of channels. Section 3.6, cell environment interactions. So cells interact with their environment by responding directly to other cells or indirectly through extracellular chemicals. So interactions always involve the glycocalyx of the cell. Remember the oligosaccharides that are either attached to proteins or the lipid components. So in A, we'll talk about cell adhesion molecules or CAMs, and in B, we'll talk about plasma membrane receptors. A, the role of cell adhesion molecules or CAMs. So every cell has thousands of sticky glycoprotein CAMs projecting from the membrane. And the functions are they anchor the cell to the extracellular matrix or to each other. Another function is to assist in the movement of cells past one another, or it could be attract white blood cells to injured or infected areas, can also stimulate the synthesis or degradation of adhesive membrane junctions, for example, tight junctions that we've talked about, or transmit intracellular signals to direct cell migration, proliferation, and specialization. Part B, roles of plasma membrane receptors. So membrane receptor proteins serve as binding sites for several chemical signals. So we have contact signaling and chemical signaling. In contact signaling, the cells that touch recognize each other by each cell's unique surface membrane receptors. This is used in normal development and in immunity. So next semester, when we look at, it's not next semester, it's when you do microbiology, you will cover immunity and B cells and T cells, and that's an example of contact signaling. In chemical signaling, there's an interaction between the receptors and ligands. Ligands are chemical messengers that cause changes in cellular activities. So in some cells, binding triggers the enzyme activation. In others, it opens chemically gated ion channels. Examples of ligands are neurotransmitters, hormones, and paracrines. In contact signaling, you have the signaling cell that is going to that has a membrane-bound signal molecule that binds specifically to the receptor of a target cell. So in this case, the cell touch each other and recognize each other. So an example of contact signaling is in immunity. Okay, so we have this or cellular immunity where we have B cells and we have T cells, and they both cooperate together to take care of pathogens. Okay, so on the left we see a B cell with a specific receptor that has responded to an antigen. And in order for this B cell to be activated and produce antibodies, it needs a signal from a helper T cell. Okay, so, and you see the helper T cell actually verifies that the antigen that is present on the surface of the B cell is in fact something that we should be responding to immunologically. So you see there's this physical contact between the B cell and a helper T cell. In chemical signaling, we have a sending cell that sends a ligand that binds specifically to a target cell that has the receptor for it. And then there would be a response. So a non-target cell would not have the receptor for the ligand, and so they would not respond to this ligand. So here we're going to look at how cell uses different strategies for sending signals. So the signal could be a hormone in the case of an endocrine gland that, so the endocrine cell of the endocrine gland is going to produce the hormone, and it's secreted into the bloodstream, and then will only bind to a target cell that has the receptor for that hormone. Paracrine signals instead are released by cells into the extracellular medium, into their neighborhood, and usually act locally. So in chemical signaling, the same ligand can cause different responses in different cells depending on the chemical pathway that the receptor is part of. So when the ligand binds, the receptor protein changes shape and thereby becomes activated. Some activated receptors become enzymes, others act to directly open or close ion gates, causing changes in the excitability of that cell. In a situation where the receptor is an activated G protein linked receptor, this one will indirectly cause cellular changes by activating G proteins, which in turn can affect ion channels, activate other enzymes, or cause release of internal second messenger chemicals such as cyclic AMP or calcium. So this here has a more prolonged response compared to the opening of a channel in the previous example. So I'll show you a figure to illustrate this point because it's not very clear just by reading this. This figure shows the binding of epinephrine, also known as adrenaline, to a specific type of receptor. Okay, so in the resting state, we see that epinephrine binds to a beta adrenergic receptor. Before it binds. Okay, so let's look at the components. We have epinephrine, then we have the receptor, the G protein, and adenylyl cyclase. As the name suggests, it's some kind of enzyme. Okay, so let's see what happens in the stimulated state. So epinephrine binds to the beta adrenergic receptor. The G protein dissociates and activates the adenylyl cyclase. And now adenylyl cyclase is going to convert ATP into cyclic AMP. And cyclic AMP is going to be responsible for increasing the heart rate, dilation of skeletal muscle blood vessels, and the breakdown of glycogen to glucose. So this happens in a stressful moment, right? When we're stressed, so more epinephrine is being produced and released into the bloodstream, and it binds to the specific type of receptor. It is going to increase our heart rate so that we can, breathe faster, and also we have more oxygen to the brain, so we can think faster, and also more oxygen to the skeletal muscles, so that we can act faster. And also we need energy for this, and then glycogen is broken down to glucose because glucose will make ATP. So that's what's happening here. So in other words, the epinephrine is your first messenger, and your second messenger is cyclic AMP. Your second, it's an intracellular messenger because it works inside the cell. And that's it for this chapter. If you have any questions, we'll discuss it in class. Have a nice afternoon.